Hello, everyone. Welcome to the show. Welcome to today's episode. Today we have our lovely esteemed guest, Nirosha Murugan. Did I pronounce that correctly?
You did. Perfect. Thank you.
Perfect. Thank you so much for being here and joining us. So, I got to do a little bit of research about some of your work, but please just give us a little bit about yourself, your work, and what you are currently doing.
Absolutely. So, hello to everyone who's listening. My name is Nirosha Murugan. I am a Canada Research Chair professor at Wilfrid Laurier University in Waterloo, Ontario, Canada. I'm a biophysicist by training with more academic training within neuroscience and cancer biology and regenerative medicine. Fundamentally, what my lab is really interested in is trying to understand how we can apply biophysical signals, so things that are rooted in light, magnetism, electricity. How do these signals interplay with biology? How do they help guide and form shape and patterns? But that's really what we're interested in. We're trying to harness these biophysical signals to understand what is health and how we can leverage it for disease. But we could talk more about that in this next hour.
So, tell me, I know that you've recently been doing some studies on, I believe it's photon emissions. Is that correct?
That's correct.
So, how exactly does that work? What got you started in doing such research?
Absolutely. So I'll start with the second question of how we started. When I was an undergraduate taking, cellular molecular biology, the gold standard way of how cells talk to each other, communicate with each other, has been well understood through molecules. There's these molecules that bind to a receptor, you have a signaling cascade, and this is what we understand as cell communication. But in those classes, something didn't sit well with me in terms of the time scale in which these cell communications happen. So that kind of led me to the physics side of things. So if not molecules, what else do cells and tissues use to talk to each other? And so through the cellular gradients, we looked at electricity. And so looking into physics, I kind of really was interested into what other physical signals do cells use. And there's a lot more biology associated with the light emission. So that's kind of how it got started. And going towards how we measure them, up until very recently, there's been quite a lot of work over the last century that cells and tissues do emit light, but only up until very recently have we have the technology to actually pick up these very, very weak signals. So that's what we're doing right now is to build out that technology to understand what these light signals mean.
And what type of technology are you using exactly?
So we use photomultiplier tubes or single photon detectors. These are detectors that pick up individual light particles. Typically, they were used in astrophysics to look at light that's coming from space and the universe from far distances. And we're repurposing that for biology by pointing them inwards into the body.
Okay, so that's very cool because last week we had Professor Pierre Humbert on from Oxford, and he was talking about using the same exact things for looking at things in the universe and so on. And then a couple weeks before that, we had another professor. I'm forgetting his name, but he does a lot of research on smoke particles after forest fires and how you can use the same thing to scan for microplastics in the brain and in the body. So all of it is very interconnected, and I find that super fascinating.
Yeah, there's a big shift in how we're trying to understand our environment and our bodies, and it's all coming together because we're looking at interdisciplinary lenses. We're putting the pieces together to try to figure out what's missing with what we
Yeah,
understand now.
yeah, it's all like a big puzzle. And what was the moment that you realized that, oh, this is something different that's maybe not talked about as much? What if you can give us a little bit of about a history about that?
Absolutely. So when in grad school, when I was looking into, the physics of life, like how does biology interact with the physical dimension and the magnetism, the what we learn in our first year physics textbooks, like what do we know? Um, when I was reading these papers, I came across the fact of a lot of Fitz Albert Pop's work, who kind of pioneered these areas showing that cells and tissue can emit light, first of all. So the fact that we emit light was very revealing and very surprising to me that in this light environment that we're in, that we're actually emitting light, not just absorbing it. Because we typically think of light as the sun and photosynthesis and things like this. Um,
Sorry to cut you off, but are we emitting light in like UV that we can't see, or is that just internally?
that's a great question. So the color of light, the wavelength of light, is actually across the visible spectrum. So UV all the way up to IR, and that's work that we've shown systematically. The reason why we can't see it is because the intensity is very, very weak. So the light that's around us is very high intense light, and then the other is looking at this thing called coherent light. So how patterned is this light that is coming out of us? There's more work that needs to be done on that end to see if it's coherent and not coherent, but there's also internal amount of light. There's light that's actually being emitted from cells and tissues from inside that is making its way on the out.
So kind of like a human form of bioluminescence.
Yes, I want to be careful about bioluminescence and biophoton emissions because bioluminescence is a very specific chemical reaction that produces light. And so we probably are aware of this in terms of fireflies or some of these aquatic
Deep sea
species.
creatures.
Exactly. So they have a very specific chemical reaction that produces luminescence or light. Photon emission, it's still light, but it's not from a specific chemical reaction. It's produced from changes in energetic states. So going back to our first year chemistry and physics, we know that our electrons are in these orbital shells that circle the nucleus of an atom. When those electrons go to a high energy state and come back down, it produces light. And so there are molecules in our body that do that, and we call them reactive oxygen species, typically associated with the mitochondria. That's what the current hypothesis right now, that where this light is coming from is probably the mitochondria.
Very interesting. Very, very cool stuff.
Hmm.
And how long have you been doing this research for?
Almost a decade now. I started this in grad school. That's kind of where I mentioned, like I was exploring what kinds of signals can we get from cells and tissue that's meaningful. We can get electrical signals. We know that from like the brain tissue and heart tissue. We can get magnetic signals. We can interact with biology using magnetic signals. And that's where I learned for the first time that in my hands that I can record light signals from cells and tissue, from human um, based cells and tissues.
And so this is always fascinating and super cool and interesting to people in the scientific community. But for the average person, why is this being studied? What are the benefits? How do you do this? If you could take people through the steps of that whole process.
Sure. I mean, there's a couple questions in there. For
Answer however you like.
Yeah,
This is we got some time.
yeah. I mean, the most exciting question is why are we doing this? Like that's, why are we investing time, money, my entire career into this? For me, it's really trying to understand what does it mean to be alive? What is life? If we're thinking about what is it to be healthy, we have all these biological markers. We have proteins, we have cholesterol, we have all these different biological markers. But there are aspects in which we live within our environment that we kind of often overlook. For example, we are bathed in this big, beautiful sun that guides a lot of our day to day cycle, but the sun emits photons and different energy patterns that our body actually needs. So that's kind of where it started. Is why do we need this? Is we need to understand outside of chemicals the physical dimension, light, magnetism, electricity. How does that interact with us? And how are we interacting with the environment? So that's the first why is, that's where it kind of started for me. And then kind of in my postdoc, I was very lucky to start working with this genius, Michael Levin, who kind of pioneered the bioelectricity domain so that cells and tissues can be patterned by electric gradients and electrical current. So that's kind of the one aspect that drew me into this area is the electricity. And when you have electricity, you have to have magnetism. There's no way to separate the two. And the one of the work that we did in my postdoc is that we showed that if you change these electrical gradients, you can start to induce really big phenomena that would seem impossible. For example, we showed that we can, after a whole limb amputation, by appropriately patterning drugs that modulate this electrical current, you can regrow a leg. So that seems very exciting.
In
And
what? How did you do that? This is not human trials, I'm assuming.
no, these are non-regenerative frogs. We started with frogs for many different reasons. They have a similar developmental pattern to humans. They're pretty aquatic, so we wanted to learn from them. But they're not regenerative. So if I were to cut off a frog's leg, they won't really grow back a leg. So it's a good monosystem. But we showed that these electrical gradients you can modulate. So the fact that you can change patterning and physiology just by altering electric gradients, it kind of brings us to the next question: is what else can we use to alter biology, to help leverage biology? And then the other dimension is it doesn't have to be treatment. Can we use this signal to listen to biology? And physical signals like light, magnetism, they're non-invasive. They can penetrate through tissue to some degree. So kind of it's the why is because it kind of opens the whole domain of understanding how biology interacts with itself and the environment, and how we can leverage that for understanding what is health and disease.
Hmm. That's really cool. I want to know a little bit more about the regeneration of limbs because you said so. Those are in frogs. Because to people listening, there are some species of animals and especially reptiles that can regenerate, like their tail, for example, or
Mm-hmm.
certain limbs. So, how does that exactly work when you're talking about using electricity within the body and all of these things? It's a little bit confusing. I would like
Mm-hmm.
to understand like the entire process, like how when you get into the lab,
Mm-hmm.
what is step 1A all the way to Z? If you
So
can condense it.
absolutely, the first step is to understand that cells have a voltage. They have a set voltage, and all the cells in your body don't have to be electrical cells like your brain or your heart. All the cells in your body are on this sort of like a gradient of voltage. And our the first question that we had when you get into the lab is, can you modulate, can you alter this voltage in a systematic way? Like, how do we change those gradients? And those are what we called ionic gradients across the cell. In the regeneration project, we knew that from building on existing literature that adult frogs, for example, when they have injury or amputation, they undergo this sort of fibrosis, pretty much like what we do when we have a scar. That's fibrosis. You're creating a scar, and scars prevent new tissue from growing. You're basically covering up a damage. So the first step that we needed to do was to create an environment that we can prevent fibrosis. If we prevent fibrosis, then maybe we can allow regeneration to happen. So how do we prevent fibrosis? And this is kind of where we turned into biomedical engineering, biomaterials. We had a great collaboration with David Kaplan using silk, and we found that silk polymers they actually help prevent scarring from happening. So hydrogels, things like this, and you probably see this in your local, pharmacy. The hydrogels are really, really good for preventing scars. So that was first step: is that can we prevent scars from happening? And we use silk to show that. The next step is we need to figure out how do we interact with the biology to help change that electrical landscape. So we could put electrodes in to jolt the tissue and put electricity in, or we can use chemicals that alter subtly this electrical gradients, these ionic gradients, and they're called ion channel modulators. For example, they change the electrical gradient. So we've identified 5 different compounds that specifically target signaling. They target various electrical landscapes molecularly inside of that tissue. And when we put it in using our drug delivery device, we saw that over time that the tissue, not only did it prevent scarring, but it started to pattern. It started to reestablish shape rather than forming a disorganized scar. So that's kind of what we measured over time. And this takes about 18 months. It's not something that happens over time. It's a very slow and patterned process.
So it was a fully regenerated, fully functioning leg?
It was pretty patterned, so it wasn't as close as the uncut, the wild type leg, but it was very patterned in the sense that it had vasculature, it had blood vessels, it had bone, it had nerves. It was functional in the sense that they could swim with it, they could use it. Another way that we measured function was sensory motor. So if we touched it, it was responsive, so that it was able to feel, sense something in its environment. So yes,
Wow.
it reestablished function.
Wow, wow, that's very, very awesome. That's super fascinating. I mean, now what's the next step in that? Like, how do you expect to be able to use that going forward? Because I know obviously to get to human trials that takes a very long time. It's a big process, but what would be the next step in this type of research? Because I want to stay on this topic just a little bit longer because it's super interesting.
Absolutely. I mean, so this is work that again I did in my postdoc at Tufts University, and so Dr. Michael Levin and Kaplan, they are continuing this work in rodent species. So they're using mouse models that are similar to humans to scale the devices so that it can be used in mammalian models. I am pursuing this as well in another dimension, but using more on the physics side. So I mentioned we use chemicals to help stimulate some of these responses. We're redeveloping these devices to use electrical, like actual electrical current and magnetic stimulation. So that now that we know that it can be done, we need to understand the signal. Like, what is the signal that we can put into the system to help kickstart the regeneration process? We know that we can prevent fibrosis. Can we put in other signals to help facilitate the same thing, non-invasively?
And how does that go through? Just non-invasive is just using, as you said earlier, using light and other things that can go through tissue.
Correct, exactly. So we've
Very,
developed
very
some
cool.
magnetic field devices. Magnetism, you don't really see them, but they have these field-like properties. They're not like chemicals that you can actually see and they're tangible. So we've developed these wearable sleeves that have magnetic stimulation coils in them that generate a magnetic field.
And you're also using this to help locate cancer, is that correct? Or
Hmm.
anything else, right?
Yes, so that's kind of where all this started is on in the cancer domain. That's where I did a lot of the work in my PhD. Um, so that's kind of where it all started. Is can I non-invasively figure out if there's cancer in a dish without looking at the chemical and genetic makeup of it? And then on the treatment side, can we use physics to interact with cancer cells specifically without having to target the healthy tissue? Yeah, so that's where it
And
all
how
started.
has that been going along?
Pretty good. We've been making quite a lot of progress on the detection side using light, and what we started in my PhD has scaled quite profoundly till in my lab right now. What I did in grad school was using these detectors. We found that cancer cells emit a different pattern and intensity of light than healthy tissue. We know that genetically and molecularly they're very different. So the physics seems to track with the molecular biomolecular landscape as well. And so what we've done since then is we've done this in animal studies where we've had animals grow very, very small tumors to very, very large tumors. If it's large, you can kind of feel it, and it's kind of pointless for early detection at that point. But it was good for us to kind of get a signature from injecting cancer into an animal where you don't feel, we don't may not know in a blind study that whether there's a cancer there in the animal or not. Using the same detectors as I mentioned, you point it towards the animal. They can freely move. There's 6 different sensors around the animal, but we're picking up the light coming off of them. Using that light signal, we can create an algorithm that says, based on this algorithm, is there cancer there or not? And that's what we have developed, and we're kind of working on refining this technology.
So totally non-invasive.
Completely non-invasive. Animal can move. They're not restricted. We have a pretty good accuracy, detection accuracy right now in the animal model. It's about 91 to 92%. And
Good odds.
yeah, I also want to mention these are surface cancer. So melanoma is where we are making the biggest progress. We have also moved towards glioblastoma, which is a kind of brain cancer, and also breast as well. So the issue that we're having right now is penetration. Like, are the light signals actually coming from deep tissue structures, or are they just surface that we're reading as like a signaling thing?
Hmm. And how, in a realistic scale, how much more time do you think this would be before it gets applied towards medicine in people?
Within surface cancers, I would say within the next 10 years, it's feasible. Our detection technology for photonics is rapidly growing and getting a lot more sensitive. Moreover than the hardware, the software has been incredibly useful and powerful. Now that we have AI and all these language models that can help pick subtle patterns and signals that we can't typically see in typical statistical models, it's become incredibly helpful. The what we're trying to be careful of is creating false positives or false negatives. So creating experiments so that we can elucidate between those two is what we're working on right now.
The whole cancer world and the cancer industry is a lot. It's a very scary topic to talk about for a lot of people,
Mm-hmm.
especially with, the ways that we have to test things and so on. The people that do this type of work, such as what you're doing, it's very interesting, and it's very selfless. Being able to find ways to help not just other people and humanity in general, but all forms of life. I mean, now we see like not just detecting of cancers, but also things such as limb regeneration and so on. But aside from those two things, is there anything else that you've learned going through this process and studying these things that you were a little bit surprised about?
I mean, to me, I still find it incredibly fascinating that cells and tissues emit light. That sentence in and of itself is pretty cool. And um, recently, we also have shown that brain activity in human living subjects that they emit light emissions that seem to be tracking what their activity is. So based on what their brain activity is, we typically use MRIs or EEGs, electrical signals, but we showed that we can detect the light coming out of someone's skull. First of all, that was pretty cool that was meaningful.
I didn't
And
even know that you could do that.
exactly. So that was pretty exciting is that the light coming out of someone's head can be measured. The caveat is, I mean, you need to have a hyper dark room. So we have a Faraday cage that is double dark chamber. So it's a dark room inside of a dark room that is lined with photo curtains. So it's extremely dark in there, so that we can pick up these subtle signals. So that's exciting is that we can pick up these light signals. And the other thing that I've learned that right now is I went into this thinking that the light that we are measuring is correlated, very strongly correlated to brain activity. And it can be, but it has opened up so many other questions that is really exciting. Is if there is light being emitted into the brain or being emitted by brain tissue and cells, how is it getting around? And if it's involved in meaningful information and processing, how are cells processing it? So right now we understand molecules and neurotransmitters when they're being released, there's a receptor and there's a place for all of these molecules. What does that mean for light? Are they using the same molecular landscape? Are there other ways that this information is being processed? So it's just opened up so many more questions for us to answer.
Absolutely. And all of this, I'm assuming, relates to quantum physics and biology, correct?
Correct. Yeah. We are working with the quantum unit, light. So it is working off of that. And when we're working with magnetic fields, there's quantum processes on radical pair mechanism, spin dynamics of these electrons. So yes, absolutely.
All the big words for everybody listening at home.
Yeah, sorry, it is jargon. It's exciting. The little electrons that we saw in our first year physics, we can alter the way that they interact within their shells. So they're energetic states. We get to manipulate the energetic states of these little electrons that are floating around.
What do you think would be the next step with this type of research, using light to detect all these things? Do you think that there's other illnesses that we could possibly detect, or maybe bone cancers or certain things like that that would be in the works?
Absolutely. I think once the biggest gap that we need to fill as a scientific community and as a community at large in general is widening our understanding of how we look at biology. First, we've spent quite a lot of time on the genomic project, proteomics, and now I think photonics, it should be like a new domain. There's a huge gap in what this light means. And I think once we understand how this light is emitted, what it actually means in biology, then this can be applied to all systems. It doesn't even have to be in humans. I mean, one application I think this could be very useful in is in the food industry, detecting contaminations in food before they go out to the market.
How
Not
would that work?
I would imagine, like, if you have shelf life of food and you may not be able to see the fungus or the mold, but you had a detection screener that's going to pick up the signals.
Okay, that makes sense.
So you can say, okay, this is, I may not be able to see it, but there's maybe something underlying there that's dividing bacteria or fungus or whatever,
Mm,
mold.
wow, that's something different. I didn't think of it that way. You can check I guess it's like getting a pre-screen for cancer. Is a
Right.
pre-screen for mold or fungus. That's very cool.
Yeah. If we find the signature of biological systems, say bacteria or mold, well, how is it any different than from cancer, right? It's
Yeah,
just we
you're
need to
right.
figure out what that language is. So the applications, I think, could be far-reaching, but there's a very big first step that we need to overcome right now.
Of course. The big step, the first initial step is always the hardest,
Yeah.
generally speaking. And so here's my question for you. This just came into my head, so I'm not sure if anybody's asked you this or thought about this, but do you think there would be a way to harness the energy created by this light, or if it's just a byproduct of us being alive and being biological creatures?
Ooh, that's a great question. No one has asked me this. The second part to that question is something that I've thought about, and is the light being emitted just a byproduct of metabolism by our mitochondria? Um, the working hypothesis that we're trying to test, amongst many other, is no. I don't think it is just a byproduct because there's some evolutionary evidence that suggests not. For example, within deep brains, like within your basal ganglia, so deep brain structures, there are proteins in there that are light sensitive. And these are proteins that are outside of your visual perspective. So they're not in your eyes, they have nothing to do with vision. So it kind of begs the question, what are these proteins and structures doing there that are light sensitive? And it's probably because we're emitting light and they probably are processing that light. So there's some evolutionary explanation for why the photon emission might be occurring. Um, so that's the first part. Now, can we harness it? That's quite exciting. And so, if we were to think really big picture and far out there, we've developed as a community some really cool technology like fiber optic cables, right? We can send telecommunication from Canada to Europe using these telecommunication cables, fiber optic cables to process information. If we understand how the light is propagated in the brain, for example, we can kind of repurpose some of this technology to attach fiber optic cables to brain tissue to propagate some of this light.
That's
Sounds
just
far
so
out there. It absolutely does. Totally understand.
that sounds so scary.
I mean, we have implants right now to help with electrical conduction, right?
Mhm.
To help with, in epileptic conditions or in Parkinson's, right? To jolt the brain to help synchronize it, or even the heart. It doesn't have to be the brain.
Yeah,
Um,
hasn't Tesla been or Elon Musk created something that can assist people that were paralyzed? What I remembered reading something.
they're all electrical based.
Yeah.
Yeah.
Okay.
And I mean, there's movement towards the magnetic side now. That's because we're building on, 200 to, I don't know how many decades of work on the electrical and the magnetic side. We're just beginning to scratch the surface on the photonic side. So blue sky stuff, but I could see it kind of getting there one day.
And then moving on from that, it is super interesting, but let's talk about some of your work at the WIS Institute, if you wouldn't mind.
Hmm.
So, what is comparative cognition?
Great question. So at the Wyss Institute, again, this is work that I did in my postdoc. Um, how this kind of might seem at a left field, like I work with regeneration, magnetic fields, all these weird things. What comparative cognition is really trying to understand what is intelligence, what is decision making across species. So when we think of intelligence, we are pretty narcissistic to think of ourselves. We are the most intelligent organism out there, but we can see
Allegedly.
allegedly, with the help of AI, I guess. Um, but there's a lot more other organisms out there that are quite intelligent, and they don't have to be one organism, right? They don't have to be an individual, like bee swarms, for example. Collectively, they work together to be quite intelligent to solve problems as a species. So, in partnership with the Wyss Institute, we aim to try to understand that one question: what is intelligence, and do you need to have a brain to be intelligent? And so we worked with a model system called slime mold. Physarum polycephalum is the technical term.
I've heard of
Um,
this study, I believe.
yeah, so many
Solving
people.
mazes, correct?
Exactly. They solve mazes, and so it asks again, how is this thing? It has no brain whatsoever. It is actually one cell. So it looks like a big venous network, but it's not multiple cells. It's one cell with millions of nuclei inside of it. So it allowed me to ask the question, how does it process information if it doesn't have a neuron or any brain structures? And how is information processed throughout this body? So the big theme of the thing that I care about the most is information. What is information? How is it processed by, magnetically, optically, and in this system? And how does it help an organism survive intelligently?
Now, what was the conclusion to that? How does this freaking thing figure it out?
So if you look in deep inside of this slime mold, it actually has fluid that moves back
Okay.
and forth. So our fluid, our blood, moves, pretty in a single direction. The slime mold actually oscillates its fluid back and forth every 2 minutes, and it's pretty robust in terms of this oscillation time. So we think it's this beat frequency fluid moving back and forth. So if we kind of go back to physics, there's fluid dynamics in there, which be able to hold information and pass this information around throughout its body.
And would you say that this fluid is sort of a precursor to a brain?
Could be. I don't know. This is not, this is a little bit outside of my wheelhouse, but there are some researchers who are looking at vascular computation. So
Mhm.
the fluid dynamics of the vascular system being able to process information. They're looking at this in plants and in humans as well.
Yeah. Well, in plants, it would make sense because plants don't have brains, but there's all these studies coming out that saying plants can detect danger or have responses, and they have, they're filled with fluids.
Mm-hmm.
But this actually brings me to something that you mentioned earlier about how there's photon receptors deep in the brain.
Yep.
Do you think that this could just be vestigial from a
Mm.
previous time?
I would say no, because
Okay.
our
Why?
biology is pretty good at getting rid of things that's useless to us, that is energy
Mhm.
inefficient. So we do have vestigial organs and like the appendix, things like this. But proteins within the brain, something that the brain is highly efficient in terms of energetics, I think evolutionarily it would have been weeded out. So it doesn't make sense that every generation or evolution that this is recapitulated unless it serves a purpose.
Mhm. Maybe we don't know that purpose. I'm not saying that I know better, but I'm
Yeah,
saying
yeah.
maybe it's a possibility because it's weird when people talk about like gene therapy, for example. Oh, we found a cure to this or to male pattern baldness or to spina bifida or we can fix this, right? Just examples. And you can adjust the genetics for somebody while they're still alive or stem cells or anything like that. But we don't know if turning on one gene to repair something will cause other issues in
Yep.
other genes or express themselves differently. So I'm just thinking in a world, you know, playing both sides here. If one thing is beneficial, sometimes it takes away from something else. We don't really know how that whole process works yet, do we?
We truly don't. We don't. This is speculation, but what we do know, and not to discredit some of the work that's been done by other researchers in this area, is that there's a reason also why the biology seems to respond to applied light. For example, infrared therapy has become widely used now. A lot of it is in the aesthetic domain, for example, but even also in repair and therapeutics. After an injury, a muscle injury, a joint injury, red light therapy has been shown to work very well.
Mm-hmm.
Some people will suggest that it's is heat. You're applying heat, so you're changing that domain. You're putting an energy into the system, but those are all terms. It's still light. The light needs to interact with biology. So I think we still don't understand how. The people are looking for like a single molecule that's going to resonate very well. But my view of this has completely changed. And that used to be my view. I used to think, hey, we need to look for a protein that's light sensitive, and that protein is going to send a signal inside, and that's the typical way that we think of this. My view has completely changed because I've started to think of biology as not just a biomolecular soup, where I think reframing our understanding as our biology as an energetic landscape, that our brain is functioning at a set point of energy, of every action potential is an energetic signal. Every heartbeat is an energetic signal that seems to be related to some sort of molecular landscape. So when we add energy into an energetic system, it's like adding more water to a container. You can kind of change the way that it interacts with the whole system. So this is all working hypothesis, but I think it's kind of helping us to reframe how magnetic fields, how would they work if there's no receptor? If how would light work if there was no receptor? It kind of helps push us this idea forward.
Fair. That's a very fair argument. That makes sense. It's nice to know that scientists can even see something and admit that this is not working, and we should maybe look at it a different way. It's a beautiful thing. The scientific hypothesis.
Yes, I mean, I wish more people were like that. And I, you asked a
In
question.
all fields, by the way.
Yes, that's true. You asked a question really earlier on. What was difficult about this? Um, and one aspect outside of the science is, I mean, maybe it's actually part of the science, is the intellectual part of this. Accepting that, maybe going in that the light emissions weren't exactly related to what we thought, but being able to be wrong about it and kind of sharing that with the community so that the community can say, and people like you can ask, but what if this? What if it actually means this? That allows us to test our hypothesis and make this field go faster and further rather than just saying, no, it can't be light. You're crazy. Light can't emit, can't be transmitted throughout the skull. So I think there needs to be a cognitive shift. And I know we're going a little off topic, but I kind of want to say this is important that there needs to be a cognitive shift in the community, especially the scientific community, to be open to topics and actually testing them. I mean, that's our job, is to test whether something is real or not.
Of course. And you can feel free to be off topic. We can talk about anything on this show. It doesn't just need to be specifically certain things. Is there anything off topic that you find or close to this realm that interests you or that you would want to maybe speak about or comment on?
I mean, the last year and a half has been incredibly transformative in this space because people are finding parallels to what we are showing. And for example, in the psychedelic community, people who are trying to understand altered states of consciousness or how do these psychedelic compounds change our brain activity, that seems to parallel what we're finding in terms of light emissions. There's subjective experience of increased light experiences in these altered states. And that kind of begs us to ask, are they experiencing more light or are they perceiving it? Is that something that they're kind of thinking about in these states?
Is that something you think you would be able to study? Just
That's
put
something that we're
somebody on
actively
mushrooms or
pursuing right now.
LSD, throw them in that dark room and just be like, okay, light emissions. Let's see what happens.
That's something that we're actively pursuing right now. So as part of this study, we've developed a helmet, a light helmet, high dense array helmet that can pick up signals all over the head now. We were only working with 4 sensors. Now we are working with 16. So we have increased our density. So hopefully we can see, and we can answer this question. When people have altered states, are they emitting more light? I would have thought yes, but based on some preliminary data, it's actually not. There's less emissions coming out of their head. So the next question is, where's the light going?
That's a very good point. I mean, you would assume it would be more light or
That's what I thought.
less.
I would have thought it would be more light, but it's actually less light. So are brain tissues absorbing that light? Is it being used for something else?
Do you think it could be a state of the brain thinks it's dying?
Well, to that point, one of our close collaborators, Christoph Simon and Daniel Oblak, and their team actually published a paper showing that dying in animals, you get these bursts of photons that are associated with that. So if we take that data, the answer would be no. That when things are dying, there should be a decay or there should be an increase. I'm not sure.
But wouldn't that just be because it's an expulsion of all forms of life leaving the body?
Could be. So my hypothesis is when people are engaging in visualization. They're seeing in their own brains, basically, if you want to say that. The light probably is being propagated. It's being used. It's being moved around. And so this hypothesis is something that I'm really passionate about, that we're working on actively right now. We are very confident that light emission is not a metabolic byproduct, I think. The second point is, if it's not, how is it getting moved around? So we're doing this light propagation experiment where we're taking axons that are aligned, that typically propagate what we think electrical information. Can it basically be looked at as a fiber optic cable? The properties of when we look at these axons can act like a fiber optic cable. So we're doing some experiments to actually prove that. If they are, then we have really good foundations to say that the light that's being emitted can get moved around in these biological structures. If
Hmm.
not, I'm not sure what this means.
And now I have a question. This actually relates again to what we were talking about with light leaving the body when something dies. Does light have weight?
Does light have weight? Light is technically massless, or very little, ultra, very little mass. So the physics of it, no. And this is where the speed of light comes from, and all of the physical derivation that we don't have to go in technical content. But I'm curious to see why you think why that question.
Why I'm bringing that up is because when people say a person or an animal dies, there's like a 2% or something reduction in the body weight of that individual. And I'm thinking to myself, is that energy or is that now light leaving the body? I don't know. It's just a hypothesis I just popped up into my head while talking to you, but I thought maybe I'd ask. You're the professional.
I mean, 2% is quite a lot of mass for an average individual. I
I
would
could
be
be wrong.
yes,
I
yes,
could be wrong on that number, but apparently there is a weight that
yeah.
disappears when somebody dies.
Yeah, I have heard this. I'm not sure if it was 2%. I have heard this, and I heard the responses as it's the total oxygen volume. You're reducing the volume capacity. It's bacteria that's dying, and because we are quite a lot of our body mass is the bacteria inside and outside of our body. Um, I would be remiss. I don't think it's light. I don't think it's the light in terms of weight.
Oxygen probably makes a lot more sense. Thinking now, it's like, fuck, that's a stupid question to
No,
ask.
not at all. I mean, we should explore this. I mean, but what does that mean? I mean, there's a lot of these subjective experiences, especially people who are working in hospice care. Having lost a loved one myself, I personally didn't see this, but I've heard a lot of people say that, close to when their loved one is leaving their body, they see light. They see a light leaving them.
Oh
And
yeah, the
so
go towards the light.
I go towards the light.
Yeah.
Culturally, this isn't something that new. Like, we've always been talking about light in our day-to-day lives. So I don't think it's an accident. But we just need to be able to rigorously quantify it and figure out what this means.
It's so fascinating, all this stuff, how everything works together.
And it's a little frustrating, I'll be honest,
Tell
because
me more. Why is it frustrating?
it's frustrating because for me, from my personal experience, it's taken a decade for people to be willing to listen. And
What
I
do you mean?
wonder, well, the the chemical communication aspect of how cells talk, that's been such a ground truth for so long that when anyone shows evidence that, hey, maybe they can use electricity to talk and it doesn't have to be the brain or the heart, maybe your skin cells can use electricity, or hey, your brain is an electromagnetic organ, or if I were to say, hey, your brain emits light, saying this 10 years ago from personal experience wasn't accepted. And I just think that, again, going back to this idea of being open and working with experts that might be in the engineering or genetic side, and we can actually make more progress faster if, we're able to work together and accepting some of these things, or at least willing
Can
to
you
test them.
actually, sorry to cut you off, but can you actually go a little bit more into how the cells communicate? I didn't get to ask you specifically how that works. I'd love to know more about that.
Sure. So we have really good understanding in cell signaling. If you look up any cell molecular textbook, you'll see a cell with a bunch of lines and arrows inside of it that looks nauseating to look at. And that's because a single molecule, when it comes inside of the cell, has a chain reaction of interacting with other molecules that then sends a signal to another one that turns it on or off, or there's a big signaling cascade of molecules. They are like a domino effect inside of cells, and they release molecules, and we call those hormones, for example, or cytokines that will trigger a response in an immune cell. And that's what we understand as chemical communication. Molecule released by cell A is picked up by cell B, molecular chain reaction inside of cell B that releases another molecule, and it's a big molecular communication. So that's well established right now. We're starting to see that cells and tissues can communicate outside of molecules. They can respond to each other even though nothing is being released. And Michael Levin has shown this in a very cool experiment where if you were to amputate an arm, you can see signaling or calcium changes, for specifically what they showed, on the opposite arm. And you would say, oh, of course, it's because there's nerves, right? You know, two arms like this, then the information goes through your spinal cord and then it comes back out again. But if you sever that spinal cord, you still see that same effect.
And how exactly did they cut somebody's hands off? What
Oh,
did they
it's not a
do?
human, it's a frog. I should be more specific.
Oh, these poor frogs. You guys are tormenting these guys.
I mean, we are very thankful for animals that are part of our experiments, and we're, taking care of them ethically as much as possible.
Is this done while they're asleep?
Yes, so they're given anesthetics to ensure
Okay.
that they're minimized pains and
The poor bastard. He goes to sleep. He wakes up. He's got no arm. He's like Lieutenant Dan in Forrest Gump. I ain't got no legs. Some
oh,
guy in a white lab coat's just poking you.
yeah, I mean, my animal models have been very helpful for, many different scientific explorations, and they give us the freedom that you can't really get with human models, right? So
Yeah, and so what would exactly happen with the calcium, with the extra calcium being put? Is it like sort of strengthening the other arm? What's
so
going on?
just to clarify, so I'll set up the experiment again. So you have an animal, um, you inject a dye into it to see the calcium changes in these two sites. You don't have to completely amputate it. You can also just injure the arm by, small damage to the arm itself. And you see a signal, an increase in calcium in the same spot on the other arm,
Interesting.
or the other side of the body. Yeah, so the other arm. So, I mean, these are just experiments to kind of showcase that. And the reason why I'm mentioning is because it gives us a sort of insight. It may not help us answer any questions, but at least it helps open the door to another question, which is, if it's not a chemical signal, what other way is the body responding to produce that kind of signal? Is it
What do you
magnetic?
think it is?
Um, I genuinely don't know. It could be electromagnetic. That could be one thing. Could be the vasculature.
Okay.
The fluid dynamics. That's a mechanical force, right? So we can't really exclude that. Maybe yeah, the mechanical the perturbation can propagate all the way to the other side.
That does make a lot of sense. So, as we are winding down, I obviously always thank our amazing guest and our amazing listeners for tuning in. But before we end anything, I do want to ask you, even if it's completely unrelated to what we've been talking about for the last close to an hour, something personal, anything fascinating, any other thing science or universe related that interests you or that concerns you that you want to talk about, please feel free to share.
Something that's been I've been thinking about that has nothing to do with science. Maybe it does actually. Um, is aviation. I'm also a pilot. I love flying. I love planes.
That's so
And
cool.
something that's been on my mind recently is like I think it's absolutely fascinating that we can get a tin can to fly and to be so high up in the sky.
You're
And now
literally in a box in
you
the
literally,
sky,
uh,
going
and
hundreds of miles an hour. It's so scary.
isn't that ingenuity came from our brains? It's
Yes.
fascinating to me. And we
I
can
told
send,
my cousin, I was telling my cousin the other day at dinner. You know, it took like 60 years to go from the Wright brothers to landing on the moon.
right. So what I've been thinking about, and is the ingenuity in spacecraft hasn't really changed. A plane looks like a plane that it did 20 years ago, 30 years ago, 40 years ago, maybe there's slight variation.
Uh-huh.
Why not? We've made so much progress in things like AI and computation. Our cars look different a little bit, you know? Um,
I guess
but
if it's not broken, don't fix it.
but we can make it better, no?
We could, I guess, but the thing is also people have the. I love how we're talking about this. This is so random, but no. But
You asked for it.
when people think of a fighter jet, they think of, oh, like what is it, like an F-15 or something? They've
Hmm.
been making those since like the late 70s.
Exactly. Not a lot of
They
progress.
look almost exactly the same.
Yeah, the Blackhawks. Yeah, it has like special materials or whatever, but the architecture and the structure is pretty much the same. So have
Hmm.
we reached the limits of aviation in our creativity? I don't know.
I don't know. I saw a meme, and maybe it wasn't a meme, maybe it was a post, and I was seeing it as a meme because my brain is fried, and it was a new like I don't know what type of craft it was, but some type of military craft. And it is in the shape and like same type of like the build looks exactly like a falcon diving. So it's like it has like a very small like pointed tip at the front and then it like rounds up and then
Mm,
like sleeks out.
mm.
It's kind of like if you put like a ball in the middle of like a very narrow uh strip and it looks super cool. And I'm not sure if that's real. US government, please let us know if that's real. I would love to see it. But I don't know. It's like innovation like that, I think is cool.
And you actually, I think, got the secret right there. I think we really need to look at nature a little bit more closely and draw inspiration, not just inspiration. I think we should copy nature. Nature has done it well over refined time through evolution. And
Billions of years,
yeah,
yeah.
really,
It
we
knows what it's doing. It works
clearly,
very well.
clearly. So I think, yeah, I don't know. That's the random thing that I've been working on and thinking about.
Amazing. We love to hear all the cool scientific stuff and random stuff in people's heads. Dr. Murugan, thank you so much for being here. Thank you so much for enlightening us on light. Haha, science. Everybody, thank you so much for tuning in. Until next time, thank you so much.
Thank you. This has been
Of
exciting.
course.